Okay, so we're just going to quickly kind of outline pre-lab stuff here for your cell anatomy lab. All right. What we're going to be doing here on Monday, when we look at these different slides, is we're going to be looking at examples of plant cells, examples of animal cells, okay, and doing some comparisons, and actually looking for particular cell structures that we've talked about in class. All right. Now, I've had a few people come and say to me, Mr. Kader, I'm drawing my amoeba and my paramecium, and I can't recognize anything in there. That's okay. All right. It was your first time. It takes a little while. And on the amoeba and the paramecium, not everything is quite as clear, okay, as it is when you're looking at an actual animal cell or an actual plant cell. Okay, when you're looking at those, things become quite a bit more obvious. Okay, I'm not sure why that is. It just is. Okay. But you do have to kind of use your imagination a little bit. I don't mean guess, all right? Like when you're looking at the cell, don't go, oh, well, that's probably this, okay? You want to look at it and go, okay, well, it's kind of fuzzy around the nucleus, and on my diagram, around the nucleus is usually where rough ER is. So that's probably what that stuff is. It looks like the nucleus has a beard. Okay, that's probably the rough ER. Honestly, that's what it looks like sometimes. Okay, sometimes it looks like the big dot in the middle has got a beard. That's the rough ER. It always hangs around near the nucleus. Okay, so you kind of have to reasonably assume what things are, okay, as opposed to guessing. Now, if you're writing down that you're labeling ribosomes, I'm going to call you on that one, because ribosomes weren't discovered until the invention of the electron microscope. You can't see those with a light microscope, all right, so don't bother labeling those. All right. Similarly, things that are typically transparent are not things that are probably going to show up in your cell. All right. But if you see a big kind of void in the middle of a plant cell, what could you assume it might be? The water vacuole, right. Okay, so you just have to kind of use your reasoning skills a little bit when you're trying to label things, right? What the cell anatomy lab's purpose is to kind of investigate, well, are there structures in common? And why would they have them in common? We've kind of talked about that a little bit from the perspective of modes of nutrition, but we also want to look at that from the perspective of cell theory. All right, what are all cells supposed to do? Second point. Carry out the basic functions of the organism. There are certain basic functions that are common to all organisms. Okay, and so as a result, they should probably have, okay, similar organelles, similar structures, things like that. Okay, these are the kind of ideas you're going to want to work into your hypothesis. In fact, your hypothesis should probably mention something about the second point of the cell theory for this lab. Okay, and the then part should probably talk about what things you think the cells are going to have as a result of that need to carry out those functions. All right, so the problem we're investigating is that cells have to carry out the basic functions of an organism, second point of the cell theory, in order to survive. Thus, they must have structures within their cells that carry out those basic functions. That's what we're looking for. All right, so in your design, identify your manipulated, responding, controlled variables. Okay, what are we looking at? Cells. Okay, are we looking at different kinds of cells? Okay, thinking that could be possibly one of the variables if we're looking at different kinds of cells. Okay, and we're looking for particular things in those cells. Okay, and we're probably doing some comparisons that are, let's say, on a level playing field or a controlled playing field, okay, that you might want to identify in there. Okay, I'm not going to give you as many as I have been because I'm kind of trying to wean you off of that a little bit. Okay, so we already talked about the hypothesis. If part should probably talk about the problem. Okay, then part should talk about what you're going to see. Okay, and part describes the experiment briefly. Okay, what kind of cells? All of these cells are one type of cell. Okay, we talked about that yesterday. There were two types of cells, not plant and animal, but they were. Anyone remember? No, sorry, it wasn't yesterday. For you guys, it was Wednesday. Nope, those are the modes of nutrition. You guys remember the words eukaryotic and prokaryotic? Okay, all the cells we're looking at are eukaryotic cells. Hint, hint. They're all eukaryotic cells. Okay, and that's important because eukaryotic cells are the ones that have all the organelles. Okay, if I gave you a bacterial slide, okay, you'd be able to see the bacteria but no detail. They're prokaryotic. They're very, very tiny. Okay, so the cells we're going to be looking at here, okay, are going to be a monocot dicot cross section, which basically means they're going to take a leaf. Okay, so they'll take a leaf like this and cut it this way, and you'll be looking at it straight on like this. All right, and as a result, you're going to see the different layers of the leaf that we'll talk about when we talk about plants later, but you're also going to see palisade cells. And palisade cells are the cells that look just like your diagram. Okay, they're rectangular. Okay, they're very fixed. They're very pushed, very crowded in there. But you'll be able to see lots of them. They're very detailed. They're quite large, right? And you should be able to see lots of detail within them. All right, you're going to see a generalized animal cell from a slide that is named stratified squamous epithelium. Basically, that's the fancy way of saying if you scrape yourself real bad, it's that red skin you see underneath. Okay, kind of right before it bleeds, the stuff that really stings. Yeah, that's stratified squamous epithelium. Right? It's full of animal cells, which are much smaller. Okay, but you'll still be able to see quite a bit of detail in them once you figure out where they are. Okay, in fact, when you look at it on high power, it's going to look kind of like dried mud, except pink. Okay, you know what happens to mud when it dries, right? It kind of cracks, and you see all the well, each one of the kind of cracks would be a cell. Right, so that's kind of what it's going to look like. And you'll be able to see little purple dots. Each purple dot's a nucleus, and that'll kind of help you to get centered on a cell in there. All right, you're also going to look at a human blood smear. Okay, it's not your blood. It's completely sealed in, so there's no risk of catching anything from it. All right, but you'll be able to look at the human blood. And you're going to make a wet mount of your own cells by swabbing your cheek like CSI. I'll give you a little Q-tip, and you can swab your cheek and smear it all over a slide. Okay, add a little water to that, drop a cover slip on like you did with the letter E the other day, except you don't have to worry about your cells flipping over and being right side up. Okay, slip it under the microscope, and you will see lots of stuff. The cells are going to be actually reasonably large, but quite transparent, because all the other cells you'll have looked at will have been stained with a dye, so they stand out more. These ones won't be. So anything that's really dark when you look at that stuff is food. No matter how well you brushed your teeth or washed out your mouth with Listerine or whatever, you're still going to have pieces of food stuck in your mouth. All right, and when you swab, they come off on the swab, and you'll see them. All right, depending on what you ate, that might be, really gross or really cool. I don't know. It's up to you. Okay, but you will see cells, and I'll kind of show you a couple of tips and hints for viewing your own cells, and that's actually the ones you'll see the most detail on once you get them focused. Okay, and then for the analysis, there's only one question. Okay, and that one question is, based on your knowledge of cell structure and your observations of the different cells in this lab, what organelles are most important for a cell to function, and which ones are specific to a certain mode of nutrition? Explain. All right, so I'm looking for kind of a paragraph answer on that one. All right, if you answered that with nucleus, yes, you're going to get like 1 out of 5. Okay, I'm looking for quite a bit more detail than that. Okay, explanations of, these were the organelles that were in all of the cells, but these ones were only in this cell, these ones were only in this cell. Maybe that has something to do with, photoautotrophs, chemoheterotrophs, whatever. Okay, that's the kind of stuff you want to talk about in that analysis question there. And then, as usual, conclusion and evaluation. All right, questions on that for Monday? This way we can get straight into the lab on Monday, look at all those cells, and hopefully have some time for you to come back in here and work with the Chromebooks and get some of that done in class. All right, Cameron. I will decide that. I'll put it up today. I'll put it up on Google Classroom today, and then I'll put a due date up. If it's due at all next week, yeah. It might not be due till like a week after that even. All right. But it'll go up on Google Classroom at some point today. Okay. Okay, reminder. What's due on Monday? Microscopy labs. Okay, remember guys, when you're drawing those lab drawings, okay, draw them out by hand, snap a picture of them, and insert that picture into your report. Remember, when you're drawing those lab drawings, you have to stipple the dark spots, okay? Don't shade. Remember, follow the instructions on that handout I gave you for a proper lab drawing. Okay, it came with your microscopy lab handout. Had the picture of the little worm, okay, on it. All right, so check that out. Make sure you follow those instructions, okay, when you're drawing your lab drawings for the microscopy lab. Okay? All right. Okay, guys, one other thing here, just to give you an idea. Here's what you're going to be looking at. Okay, on Monday, okay, this here is your monocot dicot cross section in the top left here. And you can see all of these little columns here are plant cells. Down below is what we call the spongy layer, and those are also plant cells. They just don't have as rigid a shape when they're down in the spongy layer. Okay, this is the stratified squamous epithelium here to its right. Each one of these dark pink circles, okay, so this thing right here that I'm now going to make blue, that was a nucleus before I obliterated it. Okay, so each one of these little pink dots is a nucleus, and you can see the cell shape around it. And when you get that blown up, you can actually see quite a bit of detail in each one. This is the human blood smear. And those are my cheek cells from last semester. Okay, yeah, they are pretty cool actually. I'm rather proud of them as they are mine. Okay, yeah. All right, so that'll give you some idea what you're looking at there for next week.